Hello, everyone. My name is Michael Schatz, and I'm your host for today for the Galaxy Training Academy's Meet the Expert series. Our guest today is Dr. Stephanie Hicks. Stephanie is an associate professor of biomedical engineering and biostatistics at Johns Hopkins University, where she develops statistical and computational methods for analyzing high-dimensional genomics data. She's an applied statistician working at the intersection of genomics and biomedical data science, and her work emphasizes single cell and spatial transcriptomics with a strong focus on rigor, reproducibility, and open science. Stephanie is also widely recognized for her leadership in the Bioconductor community and her advocacy for best practices in data analysis, mentoring, and inclusive scientific training, including with the Our Ladies organization. To learn more, visit her lab's website at www.stephaniehicks.com. Stephanie, thank you so much for joining us today.
Thank you, Mike. It's a real privilege to be here. Very excited to talk with you.
All right, thank you so much. What I'd like to do is talk a little bit about your past, present, and future in genomics and genomics data science. So just to kind of
Yeah.
kick things off, yeah, so just kick things off. How did you get started? Were you always interested in genomics?
I did my PhD at Rice University, and Rice University is right across the street from what's called the Texas Medical Center. At the time, it was one of the largest medical centers in the world. And so, what I did was I basically fell in love with biology
Hmm.
and how computational research and computational methods can be applied to extract meaningful information from very noisy, complex biological data. And ever since then, I've sort of just pursued that passion and followed more and more into data science, applied statistics, comp bio, and trying to figure out how to develop better tools. As genomics is really noisy and messy. So it's kind of like picking a needle out of a haystack sometimes.
It is indeed. Well, you said that you really got interested in during your PhD. I'm curious, what sort of training, what sort of experiences did you have before that?
Before my PhD?
Yeah,
Oh, um, as an undergrad, I majored in pure math, and I had two research experiences as an undergraduate, an REU, research experience for
yeah.
undergraduate. One of them was at Rice University in doing biostatistics, and then another one was at the University of Wisconsin-Madison looking at, again, biostatistics, but more related to bacterial and microbes in the gut microbiome. So, gosh, that was a really long time ago.
Okay, but it sounds like you come from, kind of a quantitative background, interest in math and statistics. You did have a little exposure to biology, as an undergraduate, but then it was really kind of, during your PhD where you kind of fell in love with the field, is what it sounds like.
Yeah, I would say one of the reasons I discovered applied math as an interesting avenue for me was in my undergraduate, we had a probability teacher, and he would consult. This was at Louisiana State University in Baton Rouge, and he would consult with casinos. And so the idea was he would have to develop these new schemes around proving probabilistically like the proportion of time that the house would win versus somebody who's playing the game would win. So he would come in with these like elaborate schemes of here's the new problem, and we're going to show you mathematically probabilities, how they are calculated. And I just thought that was wild. I was like, this is math. And so then I kind of fell in love with probability and then statistics, and that was kind of the
Oh,
next.
that's fantastic. What a very like concrete, very direct application of the field.
Yes, yes, it was. It was much better and more interesting to me than knot theory or real analysis at the time.
Did you learn about I don't know, Monte Carlo simulations in Monte Carlo?
You know, I wish. No.
And then
But
if, yeah, fabulous. And then if we sort of fast forward to kind of, kind of your current portfolio, I know a lot of your work these days is on, kind of gene expression, and you've had a number of kind of really pioneering methods for single cell biology and more recently spatial biology.
Mhm.
What's the appeal to you for studying the transcriptome?
Oh, well, during my PhD, I studied the genome. So I studied DNA mutations, specifically missense mutations, how they would impact the functionality of proteins. So I had a little bit of exposure to proteins, a little bit of exposure to transcriptomics, but mostly related to DNA mutations. And it was very clear to me when I would attend, for example, the American Society of Human Genetics or other conferences many people were talking about omics beyond changes in the genome. And so it was something I really knew very little about as part of my PhD. And I wanted to train in an area where I would really dive deep into many different kinds of omics. So during my postdoc, I was exposed to gene expression and DNA methylation and the messiness and the noisiness. And what I discovered is that the cell is very complex. And there's still a lot that we don't know. But all these different snapshots of the cell, not snail, would lead to a better representation and understanding of what's happening in the cell. And things that we can do really well with DNA methylation, we can't do so well often with other types of omics and vice versa. So at the time during my postdoc, single cell RNA sequencing technologies had just come online. And so I really just dove deep into it because it was one, fascinating to be able to profile a gene's expression in a single cell. I remember when studies had 96 cells standard as part of their data sets, or even one, just like one cell. That's how old I am. And then today it's wild, like to fast forward to today
Yeah.
to see how fast the technology has changed and how much more information we can capture from a single cell, different modalities, across time, across development, across species. It's really cool.
I noticed, for example, that some of your projects have been in like expression in different parts of the brain or sort of different disease states. What are you kind of most excited about? What have you learned from that? I don't know. What has most surprised you about maybe how the brain is organized?
I think that the number one thing I've learned about the brain is that structure and function are intertwined. If you want to understand the function of cells in the brain, you need to understand the structure. And what I've also learned is that the brain is very complex, very complex. And different brain regions have different roles and they have different mechanisms that have developed over time for why they have these roles. I actually am not a trained neuroscientist, but when I joined Hopkins, I met a set of researchers at there's a local institute right next to the School of Medicine called the Lieber Institute for Brain Development. And they have and their entire mission is focused on try to understand the genetic and genomic mechanisms for understanding neuropsychiatric and neurodegenerative and developmental disorders. So by collaboration, I've become part neuroscientist. If you attend any of my talks, I talk a lot about the brain. I'm fascinated by the brain. And I find that trying to understand the molecular mechanisms in the brain of these different disorders is actually quite challenging, but also quite fun and something that I think I hope to do for the rest of my career. So.
Well, I really like that sentiment. If I can, or if I'm hearing you correctly, I think it's been what you just said is like a very productive collaboration where you kind of bring different expertise together. I'm guessing your role in these projects has really been to drive some of the quantitative science, but you have to work with the experimentalists. You have to work with people that really understand the disease states. I don't know, how do those collaborations form?
How do they form? Um, One day, a collaborator came over to the Department of Biostats seminar and connected me and said, "Hey, we have this interesting data set. You should come over to Lieber and check it out." I guess that was kind of the very beginning of it. And so myself and a postdoc, Lucas Weber at the time, went over to Lieber. And at the time, spatial transcriptomics technologies were not commercially available. The term really had just been published only in an academic lab setting.
Yeah.
Um, and we had early access to these technologies. And what that meant was, while it's an incredibly cool opportunity to be able to profile these brain tissues with these new technologies, the computational tools correspondingly weren't available at the time. And so you're exactly right. These collaborations require a lot of team science. It's very messy and it's a lot of fun. There are many conversations that I get to ask very honest and open, transparent questions of, I have no idea what that word means. Can you please explain that to me? And vice versa. I will say though, over time, the collaborators that I've worked with are probably some of the most computational savvy people that I know. They can explain to you what a p-value is. They can explain to you what principal components are. They can explain to you what volcano plots are. So it's real fun to be able to work together with them.
Amazing. I do want to talk a lot more about the computational work in a second. But before
Yep.
doing that, maybe just for everyone on this listening in today, could you say a little bit about, what is spatial genomics? What is single cell? How does that compare to, I don't know, bulk analysis? Why would you use one over the other?
Yeah, that's a great question. So bulk RNA-seq is this concept originated 30 years ago now. I'm probably getting my decades wrong. Following up from microarrays, where we can profile all the transcriptome, the human transcriptome or any species transcriptome, at a bulk resolution. Bulk resolution means it's an average of expression across all the RNA and whatever the tissue is. So if I take a piece of my skin tissue, have many millions of cells in it. You just average the RNA across all of the cells in the tissue, and you get a readout for how much RNA for every single gene is expressed. Single cell is like that, but kind of on steroids. You zoomed in at a single cell resolution, so you can, for an individual cell, find out how many genes were expressed from one gene, two genes, three genes, all the way up to the 20,000-ish human protein coding genes. And then the spatial technologies came online in 2020, I would say, so 6 years ago now. And the reason that we're interested in trying to understand the spatial gene expression resolution, if you go back to the brain tissue, for example, structure and function are intimately intertwined. And so if I want to try to understand what brain tissue looks like or what a brain region's morphology looks like, the structure of a particular brain region, I can't just take a single cell out of its tissue and then try to make sense of it. It would be really nice if I understood what those cells looked like in a 2D space so I can understand what the structure looks like. So then I can hopefully better understand what the function of these cells are doing. So spatial lets us capture all the information in a 2D context, and it lets us capture all the things outside of the nucleus. So single cell RNA sequencing in the brain is very challenging. Most people just use single nucleus RNA sequencing because it's very hard to preserve a neuron whenever you're applying these techniques. The neurons tend to break, but the nuclei tend to be really robust and strong. And so we can identify cell types really well. But if you're interested in neuropsychiatric disorders, these are often RNAs that are misfolded or that happen something outside of the nucleus that are goes to the end of the synapse, for example, and is translating into a particular protein that is misfolded, for example, and causing misfiring of a neuron. So single nucleus RNA sequencing is often insufficient if you're interested in brain tissue, and spatial captures both the nuclei and the cytoplasm and everything outside of it. So it's really one of the best ways to try to understand neuropsychiatric disorders.
So it's a way to kind of, detect and analyze, where in the cells, even outside of the nucleus, presumably you can see like at the synapses where
Exactly.
the neurons are talking
Exactly.
to each other. You can kind of see
Exactly.
what RNA and proteins are being produced. I mean, it sounds amazing.
Mhm.
Why don't we always just do spatial analysis?
It's very expensive. That's one reason. Um, I would say also the computational tools are, well, I mean, I think the computational tools have come a really long way. When I started in the field, I would say a lot of the people just adapted wholesale kind of the tools that were developed from bulk and single cell and applied them to spatial. And in general, that works, I would say, like 60% of the time. But
Hmm.
there are a couple of standard types of analyses where that completely falls apart. And it's taken us a couple of years to figure that out. But it's very natural to take tools that were developed from one technology and then apply them in another technology and identify where things go wrong, or why things are not working in a particular way.
Yeah, okay. So maybe that's a good transition into some of the computational and sort of more statistical analysis. Like at a high level, like what are some of the major questions that you might be able to ask of, bulk data, single cell data, spatial data? What are some of the major questions there?
Yeah, I would say in transcriptomics, you're often interested in trying to understand cell types or changes in gene expression across cell types, or this new terminology, domains. Domains are unfortunately an ill-defined term right now in the field. They can be differences. A domain could be a particular region in the 2D space that's just a differences in cell composition, or there could be differences in actual changes of gene expression, or there could be gradients of gene expression. However you want to think about it, it's groups of 2D observations that you say are the same cluster, for example. So this is a very similar concept to unsupervised clustering with bulk RNA-seq or single-cell RNA-seq, but now just at a 2D resolution. So this concept of how to identify a data-driven map of what are the domains in my tissue. In cancer, for example, you often have pathologists annotating by hand regions of interest. And can I do this in an automated fashion with just the genes, for example? Or if I profile both gene expression and protein information in my tissue, can I use both modalities to come up with better domains? So that's one task that people have spent a lot of time thinking about. And
If I can jump in there.
yeah.
I'm definitely not a neuroscientist, but my crude understanding is your brain is organized into layers and they kind of, have complicated shapes. So in that organ, I imagine there's, quite a bit of variability from layer to layer, from region to region. But if you go to like other tissues, like is that spatial component of note or is it more homogeneous?
Yeah, so my first collaboration was in the dorsolateral prefrontal cortex. And for those that are not familiar with that brain region, it's one of the most beautiful brain regions to look at. There's also a lot that has been known and studied about it. So we started with this brain region specifically for that fact, because if we saw things with the technology, the new spatial technologies that didn't make sense, um, then that would be kind of a red flag for the
Yeah.
technology. So that's where we started there. And we published a data set that has become kind of widely known in the field related to the human postmortem dorsolateral prefrontal cortex. And it has these six beautiful layers of gray matter and then one layer of white matter. And that was specifically captured around the tissue folds, around the edges of the cortex in human brain. But everybody's folds are different. Your folds, my folds, everybody's folds are different. And also where the capture of the tissue was completely driven by the experimentalist. And so the fact that the tissue folds look different is completely expected, but you should expect six layers of gray matter and white matter. I can tell you for a fact that not all brain regions are beautiful or have these beautiful laminar um, annotations or these beautiful layers. There are actually many brain regions related to either in the brain stem or in the mid region of the brain where we actually know very little about the structure of the tissue. For example, in the nucleus accumbens, there are these huge gradients of expression. Nothing is layer-like. And so all the tools that have been developed based on the assumption that you have discrete groups kind of falls apart when you go into these regions where everything is just like gradients. Nothing is really discrete. It's just like high here and then low here. And trying to understand the structure in a 2D space has been really challenging in those kinds of brain regions. And also, we've been finding out by its nature, when you take a 2D tissue section of a 3D tissue block, for example, in human brain, you're often ignoring the Z plane. And in certain brain regions, the Z plane is actually really important. So, yes, I would say to summarize, not everything is beautiful layers. Some things are incredibly messy. And then also, it's more complicated than just a 2D resolution. It turns out 3D resolution is really important for many domains.
So I guess, this is a very crude analogy, but if I don't know, cut open my laptop, the CPU would have sort of a different wiring diagram than the
Mhm,
GPU, than the RAM, than the
mhm.
disks and everything. So even in our own brains, different parts have like substantially different wiring diagrams to them.
Absolutely. Absolutely. Yeah.
Okay. You mentioned that it's expensive. Like, what drives that cost?
That's a great question. I would, so as someone who doesn't purchase these technologies myself, I'm a completely dry lab, but as somebody who works closely with experimentalists who are purchasing these technologies, I would say a little bit of it is probably commercially driven. So there are some companies that offer products that a lot of people adopt very quickly. And then there are a lot of new companies who come online with new ways of profiling gene expression or proteins or different modalities across even a Z plane, for example. So I would say very much depends on the commercialization and the availability of the tools at the time.
Hmm. Okay, so and then you mentioned that, at a sort of quantitative computational level, the sort of domain detection is related to like unsupervised learning. Could you say a little bit about that process? And I don't know, what are some of the pitfalls along the way?
For unsupervised clustering,
Yeah.
yes. So, for example, in this new brain region, or this it's not a new region. It's just I'm most recently working with this brain region, so it's new for me. The nucleus accumbens, it's very clear that if you try to discretize something that is continuous in nature, you're going to get bad results or results that are inconsistent depending on what your random seed is,
Yeah.
essentially. We tried to publish this both ways. We pre-printed a paper where we did unsupervised clustering, assuming discrete groups, and then we also tried using non-negative matrix factorization and topic modeling and other more continuous approaches. And then the reviewers actually came back and said, "Why are you doing this both ways? This is confusing." And we said, "We assumed that you would want us to cluster." And then it turns out that was not the case. So I've never seen a paper not try to cluster before with spatial data, but apparently we're going to try. So I
Okay,
would
so I
say
guess
that's a big problem.
yeah, and I mean I'm aware of a few like dimensionality reduction techniques. I guess in some sense like non-negative matrix factorization would be an example of that. I don't know. Are there any others that people should be aware of in the field, either for spatial or single cell data?
Oh, for unsupervised clustering, I
Yeah.
would say a lot of people use deep learning models now. That tends to be a really powerful way to embed the data so that you can do many downstream tasks. Some of it is unsupervised clustering with the latent embeddings, for example. Some of it is annotation. Let's say if you wanted to annotate a new data set, what the spatial domains are. There are many different downstream tasks, also integrating across multiple donors. So,
Hmm.
for example, these days we're generating data sets with like 100 donors. So, 2D tissue sections, one from each donor. And we're interested in trying to understand the molecular differences between, say, a neurotypical donor and someone with schizophrenia,
Yeah.
which is a very messy phenotype to begin with. But also, there's a lot that's known in the literature from gene expression specifically and protein information. So we're interested in trying to figure out how do we aggregate the information across the donors to make sense of it. Or if we take our 3D tissue block from one donor and take multiple tissue sections along the donor, that one tissue block, can I reconstruct what the tissue block looks like in 3D, given only 2D snapshots along particular points of this tissue block? I would say some of those are some of the more challenging tasks, how to aggregate across donors or how to aggregate across multiple tissue sections within a tissue block to reconstruct a 3D tissue block
Yeah,
with
it
little
sounds
snap.
really complicated. You mentioned that there's deep learning and techniques where you kind of learn these embeddings. Could you say a little bit about how that process works?
Yeah. So it very much depends on your architectures, as with all things in deep learning. I would say people typically take the spatial coordinates of a 2D tissue section and embed the data in a graph. So they use the spatial locations for each observation in a 2D space, and they say, for example, what are my k nearest neighbors? And then I want to build an adjacency matrix with information saying, I know that these two positions in my 2D space were connected locally. And then I can build edge weights between nodes corresponding to, for example, how similar the gene expression is or how different the gene expression is. It's also a form of data reduction. So if you take a very high dimensional space with lots of observations in 2D and convert it into a graph, this is not new, but it's a great way to reduce the size of the data. And then from there, I would say most of the literature is using graph-based architectures, similar to the way that many deep learning algorithms use. And a lot of what is different, I would say, between machine learning that uses these architectures and genomics is that our data tends to be very unsupervised. And
Hmm.
so we're using, for example, contrastive self-supervised learning as a way to try to improve the embeddings or clean up some of the latent embeddings in these architectures without the use of explicit labels for something like cat, dog, the way that we do think about them in machine learning.
But it seems like it's really advanced from, I don't know, single cell. For a while it was very popular where figure one of every single cell study was like a U map of all of the expression data. So
It
it's like,
still is. I think it still is. It's in there somewhere. It may not be figure one.
but it just
Yeah.
seems like these kind of graph-based approaches were sort of anchored in their spatial positioning, but then, you also leverage the expression data at the same time. It just seems a lot more powerful to kind of pierce through all the complexity in the cell composition.
It is, and even today, I would say, or so we're moving past, autoencoders or variational inference. We're moving into transformers and architectures that take, for example, a single cell's embedding and then translates that or embeds it the way we think about natural text.
Hmm.
So, for example, if we think about words in a document, you can say what's the most frequently used word, and you can rank the frequencies of the words. It would be similar to the genes, like what is the most highly ranked gene, what is the most second highly ranked gene, and so forth. And so you can then convert the gene expression, which is continuous in nature, to a set of ranks
Huh.
that a transformer architecture, for example, can encounter. Now, you could say, I just spent a lot of money generating my very high dimensional continuous data. Does it make sense to embed my data in this way? Maybe not, but I think the architectures need to be adapted in terms of how do I embed gene expression data in something that was built for natural language. I can tell you if I know what the most highly expressed genes are, I could probably guess what the cell types are. And that's what we can do really well with architectures is tell us what the cell types are. And that's a function of the way that we're currently embedding data. But a lot of people are thinking about how to embed the data more cleverly. So it's very exciting time in this field.
In natural language processing, there's often this like pre-training phase where you do some version of like mass token prediction, where you like hide some of the words and then
Yeah.
have it predict what's in there. I mean, is
Yeah.
that by analogy what you're doing here? You kind of hide the expression of certain genes and then you kind of encourage it to learn to predict those based on the expression of others.
Exactly. Yep, exactly. Yeah, there are many um, ideas that are being borrowed directly from natural language architectures to genomics. And some of it does work kind of out of the box, and then some of it doesn't. Understandably so.
Yeah. I mean, I guess there's a great tradition of bioinformatics stealing from natural language processing, going back to some of the original alignment algorithms and HMMs and, kind of other
Absolutely.
frameworks for sort of this sort of probabilistic analysis.
Absolutely. Yeah.
Um, I've read about something called Simpson's paradox. Maybe could you explain that to the audience today?
Oh my goodness. Um, okay. This has been a minute. So Simpson's paradox is this idea of if you see an association between two things, um, then you might ask if there is or often that relationship can be explained by something else that is often considered as a confounder.
Yeah.
And that if you take into account that confounder in terms of how it fits into the relationship between understanding X and Y, it turns out that the relationship between X and Y is not actually real when you adjust or kind of condition on this third variable. So Simpson's paradox is widely used in genomics or genomic data analysis or genomic data science because often when you're trying to understand the relationship between two things, it can be explained away once you take into account that third variable or that third confounder, if
So
that
what would
makes
that confounder be? With like different cell types or different brain regions, or what does that
Um,
often?
could be, for example, like a batch effect. Could be many different things. Yeah, it could be cell types, many different things. Cell composition.
A version I've seen of this is like if you look at all the data, there may be a positive trend, but then
Mhm.
once you sort of break it up into the groups, each group has a negative trend.
Negative trend. Yeah.
It can totally reverse your interpretation of the relationship once you kind of pierce through this.
Yeah. Yeah, I would say that's one thing to keep in mind when you're interpreting this kind of data, that this is a snapshot of what was happening in the cells or the tissue at one point in time. We don't
Yeah.
have information across time typically, unless it's intentionally designed in an experimental way to do so. And we're not making statements about causality. We're making statements about associations that we see. I will say when you start to bring in other data modalities such as chromatin conformation or ATAC-seq, trying to understand the things that regulate gene expression, that truly we know regulate gene expression, that becomes really cool when you can start to see how nucleosomes are wrapped up in different stages of development or something. So.
Excellent, excellent. So you mentioned that this sort of domain or sort of clustering is one major task. Could you say a little bit about some of the others? You know, a popular one is like differential expression. What's the sort of the state of the art there?
Yes. So I would say differential expression is this concept to understand what are the differences in genes between two groups, for example, a neurotypical group and schizophrenia group. And gene expression or differential gene expression has been around since microarray days, or probably before then. Anytime we've been able to measure tens or hundreds of genes at a time. The state of the art is to typically aggregate information using spatial transcriptomics. I would say that the state of the art is to aggregate information across observations or spots or bins at a donor underscore cluster resolution. And what do I mean by that? I mean, because we don't have a way to really integrate information across donors, what we do is we do this very this simple trick called pseudo bulking, which is to say, if I know all these observations in domain one, such as layer one in my brain tissue, are supposed to be the same, I'm going to sum up all the genes across all the spots in that one domain for that one donor. And then I'm going to do the same for all of my domains for all of my donors. And then from there, it's kind of like bulk RNA-seq, where we can do standard differential expression analysis using either linear models, linear mixed models, generalized linear models, all the standard ways that people have developed very robust tools
Yeah.
for a long time, and identify what genes are differentially expressed. I would say another advantage is because you want to do this is because the sparsity. So at a single cell resolution or at a even a subcellular resolution, you don't get that much RNA.
Yeah.
And so it's very sparse, and that's to be expected. So aggregating information at this way is kind of nice because then you can borrow robust infrastructure that has been developed for a long time in Bioconductor and Python and beyond. So
When you say sparse, if there's, I don't know, 20,000 protein coding genes in the genome, you know, in a typical spatial readout, like how many genes might you see activity on?
how many genes? Sometimes like a couple dozen.
Wow.
I mean, it can be very sparse, and the total number of counts for that cell could be anywhere between like 100 and 1,000,
Wow.
distributed across, 50 genes, for example. And that's kind of normal. I mean, a cell doesn't necessarily need to be expressing all of its transcriptome at a given point in time. In image-based technologies, we are actually limited to the number of genes that we can profile. We can only profile, say, 200, 300 genes at a time, typically. So you're not seeing the whole cell. You're seeing a panel, a very subset of the cell, molecular readout, and trying to make inference or trying to identify differences is very challenging.
Presumably, hopefully the panel is well selected so you can get kind of really informative genes in there.
It is, it is. But a lot of the tools that we use for differential expression, such as this concept of trying to calculate a library size, depend on having the full transcriptome to be able to aggregate information across all the genes.
Hmm.
And when you only have 200 genes, you could ask, is that a fair representation of the library size? And it turns out it's not true. It's actually a very bad representation. So, yeah.
Interesting. Well, you mentioned tools, and earlier you mentioned Bioconductor. Could you tell us a little about like what is Bioconductor?
Yeah, Bioconductor is a project that has existed for decades now. And the goal, I like to think about it as kind of the cousin of CRAN for biological data. So CRAN is this well-known repository of R packages that's been around for a long time. I don't remember the origins or how long CRAN has been around, but it's a great way to contribute packages, R packages specifically, that are built on a regular basis so that you can have somebody install them from this repository. In contrast, you could probably just put them on GitHub, but there's no repository of GitHub repositories. And
Hmm.
so it's really hard to have this information just kind of spread across the web. So there's this one place where you can generate or go to install R packages if you're interested. Bioconductor is kind of like the cousin project to that, but that's focused on biological data or high dimensional or high dimensional or high throughput biological data. I would say its origins were definitely starting around bulk RNA sequencing from microarrays. And a lot of the original developers and contributors to Bioconductor were statisticians or computational biologists who developed a lot of the foundational models that we use today for differential gene expression. Bioconductor still is doing very well today, but I would just say it's expanded out in terms of the kinds of data types that one uses and one can install packages from. And that's because of the technologies that have changed over time. So, for example, we didn't have spatial transcriptomics starting in 2020. Now we do. Now there are dozens of packages or hundreds of packages probably that have been developed for this data. So Bioconductor is a really great way if you're interested in using packages or interested in contributing packages. It's an open source and an open development project, meaning that you can see the code and you can also contribute to the code and make it better depending on what project you're working on.
What do you see as the advantages or maybe disadvantages of that open development?
Um, So the project is quite big and broad, I would say. And I would say the advantages are the community makes it what it wants in terms of if you want to see more packages of something, then you can contribute to it. You can start working groups for it. But then on the other hand, as the community grows, there still needs to be core maintenance of packages that are dependent on a lot of the different packages. For example, there are core packages of Bioconductor that are maintained by developers who are paid by the project, often out of federal funds, for example, to maintain those packages. Because if those packages break, then dozens or hundreds of packages across Bioconductor break. And so they have to be maintained over time. And so I guess one challenge is what are the core set of packages in Bioconductor and how do you choose which ones get to be maintained? And that kind of changes over time because you can imagine as technology changes, maybe you want to have more maintenance to newer technology so that people really have the infrastructure to build it out. Um, yeah, I would say that's a major problem.
Well, what are some of the upsides then?
Okay, so the upsides are that if you want to contribute a package to it, it's very easy to do so. And it's very clear in terms of how to do so. It's also the kind of thing that once I see somebody has published a paper and cites a bioconductor package, I know that package is going to work. There's something to be said for having a bioconductor package that you had to go through a high level of
Yeah.
review in a robust evaluation of that package that you know it's going to work. Often what happens when whenever I evaluate packages just on GitHub, it's very hit or miss about whether or not it's installable. And that's not the case with Bioconductor. So it's actually quite nice.
Yeah, I will
Very
say
capable.
in Galaxy, we've specifically written wrappers for many of the Bioconductor packages so they're available for exactly that reason. We have a lot of confidence that they'll work correctly and give you kind of high quality results.
Yeah.
Uh, Also, just so you're aware, and so everyone's aware that inside of Galaxy, you can run a so-called interactive tool where you can run either like Jupyter notebooks or even a full-fledged version of RStudio where you can get access to Bioconductor there as well. One question I always have is, is there a tension about, and I know Bioconductor's origin is sort of, like you said, the sister to CRAN and sort of the R environment. Um, uh, how is that facing kind of modern genomic data science where, I don't know, Python and Rust and other languages are become really popular as well?
Yeah. That, oh my gosh, I think I don't have enough time to elaborate on that. I'll give you my take. I think it's a really fundamental problem and an issue that Bioconductor has been thinking about at least for 5 to 10 years now, very actively. I can tell you some strategies that people have used to try to address this, and I can tell you my thinking on it. So, in terms of strategies, people have been, developers specifically, have been building packages to make the interoperability, for example, of objects between R and Python or code between R and Python to be more seamless. So you could imagine a world in which no matter what package or no matter what language the package was developed in, if I have some kind of core infrastructure that can be stored on disk somewhere, but can be loaded into Python or loaded into R, and then can go in between the languages interoperably, that's kind of a win-win for everyone. So trying to lower the barrier for how to make the infrastructure and the code translation between these two things easier is a huge, huge goal of Bioconductor, but also me personally. And the reason I'm motivated for this is because a lot of my research has been is, I would say, both in Python and in R. And so I see those pain points very acutely.
Yeah.
And so I'm sure many others are seeing this as well. There are packages such as Basilisk or Reticulate within R and Bioconductor that have been developed to try to improve these things. There's also packages around just data infrastructure. You could imagine storing data in a single object and then being able to read them into R or Python with all the bells and whistles that you want in a very seamless kind of way. That is happening, but I would say it's not often so funded by federal support because it's not as often as exciting as developing a new deep learning algorithm, but it's absolutely necessary. It's
I completely
absolutely necessary. Um,
agree.
yeah, it's a big tension. I actually don't have a preference anymore about R or Python. I use so much in my day-to-day work of both. Um, and these days, I actually have students I ask them to develop, if they develop a package in one, I ask them to use a large language model to translate it into the other language and then submit both one on PyPy and one on Bioconductor. So,
Yeah,
um,
that's becoming quite, effective, right? Where, very, very quickly you can do that sort of translation from one language. It's not always perfect, but wow, it's really powerful when it works.
yeah, I'm actually thinking about having this as an example in one of my classes in the fall for a particular like homework assignment. I can keep you posted how that goes.
Well, in the last minute here, what I like to do closing out, we're part of this training academy. I guess I was wondering if you have any advice for students that are entering the field but are not really sure how to get started. Are there any, I don't know, resources you can recommend on, how to do different types of analysis or other resources?
Yeah, I would say learning how to analyze genomic data is a very challenging task and a daunting task if you've never done it before. So I just want to like normalize that feeling if you're feeling overwhelmed. A lot of development has gone into developing these packages and putting them all together. And I would say that the best way to learn is to pick up a data set
Hmm.
and then try writing functions, either using a chatbot and say, what if I need to analyze or create a principal component plot? How can I create a principal components plot? and then try changing the parameters, try changing the inputs and the outputs to see what changes. I would say you don't want to just use the functions that come out of a large language model and not explore them, because then you're really missing the point. And I would say another great way to get into this field around analyzing genomic data is to use resources that have been developed that incorporate more than just the codes itself.
Mm.
So I find sometimes ChatGPT or Codex or Claude Code is really good at explaining, or is really good at telling me what the code should be, but then trying to understand the why behind it is not often clear. And so trying to seek out resources for understanding why this plot looks strange or why this code isn't working is I find to be one of the harder parts, but also one of the more satisfying and validating parts to being able to understand and translate those ideas across different data sets.
Fantastic. Are there any, I know within Bioconductor, there's a lot of like vignettes and help packages. Is there any in particular that you can recommend?
Yeah, I've written or contributed to two books now. So one of them is for single cell and one of them is for spatial transcriptomics. Both of them are called orchestrating data analysis for single cell transcriptomics or orchestrating data analysis for spatial transcriptomics. Both of them are built weekly on the Bioconductor build system, so you know they work, and it has lots and lots of chapters, lots of vignettes. Lots of workflows end to end explaining both the code but also the why that I was talking about for why something should be used or why something doesn't work the way you should expect it to be. I'm happy to link to those resources afterwards.
Excellent. Well, I think within our training academy this week, we have like a few tutorials to get started, but it sounds like if you're looking to do a really deep dive, I think those are the so-called OSCA resources. Is that right?
Yeah, that's it.
We
Oscar
should
and Asta.
we should go. Please just go Google them. Stephanie's very easy to find online. These papers are very easy. So, Stephanie, thank you so much. It's been a wonderful discussion today. I really look forward to all your research for many years to come. Thank you so much.
Thank you so much, Mike. Thank you everyone for your attention, and please do reach out if there's anything I can help with.
All right. Thank you, and good luck on the academy this week.
Bye.